Hello students, welcome to our online video tutorial class. I think you have all seen the video I have made on the chapter Plant Kingdom, and specifically in that chapter I have covered up to bryophytes before the vacation, and today I am starting from the pteridophytes, and then we will be discussing about the gymnosperms, angiosperms, and at last the life cycles or alternation of generations in plant. So I think who has seen the previous video, they have understood properly up to bryophytes. So let's start the pteridophytes, that is the third category or the class within the plant kingdom. First of all, we have to know about the characteristic features or general characteristics of the pteridophytes. Pteridophytes, the word if you see properly, you will get that pteridos, that means fern, and phytes means plants. That means pteridophyte means fern-like plants. So whatever the general features can be found in case of pteridophytes, the pteridophytes include horsetails and ferns. Fern is the representative plant within these pteridophytes. Horsetail is also another representative plant species or plant category present within pteridophytes. Keep it in your mind, these horsetails or ferns categories which are available in our environment, they are mostly used for the medicinal purposes, and sometimes or frequently also you can say they are used as ornamental plants, okay, because the leaves, the other structures present in the plant body, those are very much lucrative. So for that reason those parts we can use for decoration. They are the first terrestrial plants which have produced the vascular tissue, that is xylem and phloem. But this xylem and phloem, that also is not present entirely within these pteridophytes, because the xylem is not having the vessel element and the phloem is not having the sieve tube. So, or the rather you can say the companion cell. Keep it in your mind, they are commonly found in the cool, damp, shady places, though some can grow in the sandy soil condition also. The main plant body which is commonly found or available in the environment, they are sporophytic, and that is properly differentiated into root, stem and leaf. Mostly these plants are perennials, that means they can survive for multiple years, okay. Sometimes they can be annual also. Leaves can be of two types. Smaller leaves are also present, which are known as microphylous, and larger leaves are also there, those are commonly known as megaphylous. They are having sporangia, that is the sac-like structure within which the spores are stored and they are getting matured. If that sporangia is present or located on the stem, then it is known as collen, and if those sporangia or the sporangia present on the leaves, then it is known as foliar, okay. So these two terms please keep it in your mind. If the sporangia will be present on the stem, then it is known as collen, and if they are present on the leaves, then it is known as foliar. Next point is that leaves bearing sporangia, that is called as sporophyll, because sporangia or the spore sacs are present on the below surface or the under surface of the leaves. So on that time that specific leaf or leaves will be commonly known as sporophyll. Sporophyll can be of two types. One is megasporophyll, another one is microsporophyll. Microsporophyll means it is very easy, smaller leaf with spore sac, because microphylous means smaller leaves. So if the smaller leaves are present with the spore sacs, then it is known as microsporophyll, and if larger leaves are present with spore sacs, that is known as megasporophyll, okay. So microphylous is the smaller leaves, and microsporophyll means smaller leaves with spore sacs, and megaphylous means larger leaves, and megasporophyll means larger leaves with spore sacs present under surface. So I think these all of the points whatever I have discussed in this slide, it is very much clear to each and everyone. The basic features or the general features I have discussed in the previous video also, when we have discussed about the comparative study among Thallophyta, Bryophyta and Pteridophyta. But after that also some special features are there actually. So those special features are written actually here. So for that reason I am sharing these points with you. Next point is that sporangia, that is produces spores, but meiosis from spore mother cell. Obviously the spores will be produced from the meiosis by the help of meiosis cell division, and that is obviously from the spore mother cells. Okay. Spore germinates to form free living, mostly photosynthetic, thallus-like structure. That is gametophyte, and that is called as prothallus. So the thallus-like gametophyte structure, which is photosynthetic, which is mostly found and which is free living also. So these features we have to keep it in our mind. That is commonly known as prothallus. That means the definition of the prothallus we have to know properly. Male and female sex organ they are having, and the male sex organ is commonly known as antheridia and female sex organ is commonly known as archegonia. So these two terminologies also we need to keep it in our mind, that antheridia is the male sex organ and archegonia is the female sex organ. Next point is that pteridophyte sex organs and process of fertilization. So sex organ already we know, antheridia is the male sex organ, archegonia is the female sex organ, but how the gametes produced by these sex organs are performing the fertilization process. Let's see. First, in the left hand side you can see the antheridium, from where the antherozoids are producing, and antherozoids are actually further getting developed or matured into sperm. And in the right hand side you can see the archegonium is present, further which is containing the venter, and that venter is further producing the egg, that is a female gamete. Now the sperm is start moving towards the egg, and that is through the water flow. Just through the water flow the sperm will be moving towards the egg, automatically the zygote will be forming after fertilization. Okay, so this is overall the sex organs as well as the process of fertilization which is taking place within the pteridophytes. Same kind of fertilization process you have also seen in case of bryophytes. So same mechanism is followed by the pteridophytes also. Now we are just moving for the next slide, that is the types of spores produced by the pteridophytes or ferns or horsetails. Actually we all know that the fern or the pteridophytes are producing sometimes one type of spores, and on that time those kind of pteridophyte plants are known as homosporous plants because they are producing same kind of spores. Sometimes even those specific plants, pteridophyte plants I am talking about, they are producing different types of spores. One is smaller spore, that is microspore, another one is larger spore, that is megaspore. So when they are producing these two types of the spore, microspore and megaspore, so obviously on that time they will be considered as heterosporous plants because they are producing two different types of spores. If you come to the example, homosporous pteridophytes will be Lycopodium, Pteridium, these are the examples, and in case of heterosporous pteridophytes will be Selaginella, Marsilia, these are. Next point is that the megaspore germinates to form female gametophyte. Always it is a common sense, megaspore will be the female spore, so it will be germinating further to produce the female gametophyte, and the microspore, that is a smaller spore, that is mostly germinating to form the male gametophyte plant body. The female gametophytes in these plants are retained on the parent sporophytes for variable periods. So keep it in your mind, this is a very important point. It is told that the female gametophyte which are present in these plants are present on the parent sporophytic plants for variable periods. Okay, and the development of the zygote also into young embryo takes place within the female gametophyte. That means on the parent sporophyte, whatever the female gametophyte is present, within that the zygote formation and further the development of the zygote into embryo, entire the procedure is taking place. So this is actually the type of spores. Based on that, the classification of the pteridophyte, that is homosporous and heterosporous, and after that how the gametophytes are taking birth and how those gametophytes are producing zygote and embryo, that is actually told in this slide. Just moving for the next slide, and in the next slide you can see properly the figures of various plants belonging from the pteridophytes. Here first of all you can see the Selaginella, that is a representative plant of the pteridophyte. Equisetum or horsetail, it is looking like a tail of the horse, that's why the name has been given like this. This is the common fern, what you can see everywhere. Salvinia, that is also aquatic plant, but it is also actually a pteridophyte. Okay, going for the next slide. In the next slide we will be discussing about the classification of the pteridophyte. We only need to know the names of the various classes under pteridophyte. Characteristic features and all these things we don't need to know. First one, pteridophyte is subdivided into 4 different categories. One is Xylophyta or Xylopsida. Second one is Lycophyta or Lycopsida. Third one is Arthrophyta or Sphenopsida. Nowadays we are telling this Sphenopsida or Equisitini. Filicophyta or Pteropsida. So these are the 4 different classes or categories under Pteridophyta, or subclasses rather you can say. 1 or 2 examples we have to keep it in our mind, that within Xylopsida, Xylotum and Rhynia both are present. Within Lycopsida, Lycopodium is present. Within Sphenopsida, Equisetum is present, and within Pteropsida, ferns are present. That means ferns are belonging from the Pteropsida, Equisetum is belonging from the category Sphenopsida, Lycopodium is belonging from Lycopsida, and Xylotum is belonging from Xylopsida or Xylophyta. So this is actually the 4 different classes with their examples written here. So only we have to know these class names and one example of each and every class, nothing else. So with this slide, the details about the Pteridophyta, their characteristic features, how they are reproducing, how they are performing sexual reproduction, how they are producing spores, the types of spores they are producing, even their figure and classification, all these things are getting covered. Now in the next slide we are entering for the next plant class, that is Gymnosperm. Keep it in your mind, Gymnosperm word means gymnos, that means naked or exposed, and sperm, the word may actually come from sperma, that means seed. That means in case of which plant the seeds are not covered by the fruit, rather they are present in naked condition. So those kind of plants are all placed within this category, that is Gymnosperm. So let's go for the next slide, and this is the next slide, the general and special features of the Gymnosperms. Keep it in your mind, they are not bearing any flower, so they are non-flowering plant exclusively. Gymnosperms include medium-sized or tall trees as well as shrubs. They are seed-bearing plant, but the seed is present in naked condition. It is present in exposed condition, not covered within a fruit. Keep it in your mind, vascular tissue is present, xylem is present, and that is containing trachea and xylem parenchyma, xylem fiber and vessel. Both are absent. So keep it in your mind, trachea and xylem parenchyma will be there in the xylem, but xylem fiber and vessel both are absent. Keep it in your mind, they are called as soft wood. Why? Because the xylem parenchyma and trachea, these 2 structures are present, that's why the wood which is formed by them, that is the soft wood, that is not the hard wood. Within the Gymnosperm there is one specific type of plant, that is cycads or cycas type of plant. They are producing the cones. Cone is rather a reproductive structure which is present within the them. Cone can be of 2 types, one is male cone, another one is female cone. So in case of which plant, this male cone and female cone obviously present, but in the 2 different plant body. That's why they are the dioecious plants, because when we have already studied in the first chapter that monoecious plant means those plants having both of the sex organs, but dioecious plant means those plants which are having not both of the sex organ, rather the sex organs are present in separate body. So that's why they are dioecious plant. They are having taproot system, not the adventitious root system. That means the root system has developed a lot. In algae there is no root, no rhizoid. In bryophyte, rhizoid like structure has come. In pteridophyte, that root structure is present, that is adventitious root, and in Gymnosperm it has come to taproot system. And stems are also unbranched or sometimes branched also. So both type of stems can be found. Going for the next, that is the root. Within the root, the algae can present symbiotically. On that time, the root will be called as or considered as coralloid root. And sometimes the fungus can be also present symbiotically on the surface of root. And on that time, the root will be considered as mycorrhizal root. Keep it in your mind, the leaves are also can be of two types. One is foliage leaves, another is scaled leaves. That means in case of some of the leaves that is only looking like a leaf, foliar structure is present. That's why their name has been given foliage leaves. Another leaves are containing so many scales or needle-like structure. So that's why they are known as scaled leaves. Needle leaves may be simple or compound. So the needle leaves, needle containing leaves which are present, they can be simple leaves or they can be compound leaves like tamarind. So this is overall the basic features as well as the special features of the Gymnosperm. We are moving to the next slide. There are also some features are given. Exclusively if you see their ovule, that is orthotropous ovule. That means we all know the micropylar end of the ovule will be facing upward. That is a straight ovule commonly told. Sexual reproduction, that is oogamous type and syngamous type, both kind of happen. Gymnosperms are well adapted to the withstand extreme environmental condition. That means during any kind of natural calamity they can survive. Gymnosperms are having thick cuticle and they are having also sunken stomata. That means the stomata which is present quite in depth, and due to presence of the sunken stomata, obviously that will be reducing the water loss, because during the transpiration when they are losing the water, that is not maximum, that is very minimum, because their stomata is sunken as well as the cuticular layer is there. Gymnosperms are exclusively heterosporous. There is no option of being homosporous like pteridophytes, and obviously then they have microspore and megaspore. In Gymnosperms, the male and female gametophytes do not have an independent free living existence. There is another point. In Gymnosperm, the male and female gametophytes do not have the independent free living existence. Independently they can't survive. They have to depend on each other for survival. So in these two slides we have discussed about the basic features as well as special features of the Gymnosperm. Now we will be going for discussing about the male and female cone structure, which are only found in case of Gymnosperm, not in any other plant species. First you can see the structure of the male cone. Keep it in your mind, their location is actually they are originating below the apical bud. Their centrally located axis present, and that is arising from the main stalk. They are having the microsporophylls which are actually arranged in a spiral manner, and that is surrounding that axis or the stalk. Each microsporophyll has a sterile apex and two sporangia at the ventral side of the microsporophyll. So microsporophyll which is present actually spore bearing leaf, spore sac bearing leaf, they are having sterile apex as well as they are having two sporangia which is present at the ventral side of the microsporophyll, and those are yellow in color. Okay, so you can see here this is the ventral side of the leaf or the microsporophyll where yellow color sacs are present, that is the spore sac, and within that spore sac like structure pollen grains are also present. Each of the sporangia contains microspores which are three layered: exine, mesine and intine. So actually they are actually told spore sac, but it is actually like a pollen sac which is found in angiosperm plants. So here I want to say that they are having spore sac or pollen sac whatever. In case of microsporangium, that means the microspore containing spore sac, they are three layered. What is the outer layer? That is exine, the middle layer that is the mesine, and the internal layer that is intine. Okay, now just going for the next type of the cone, discussion about the cone, that is female cone. Keep it in your mind, 1 to 4 female cones will be there in each and every shoot. Here microsporophyll will not be there because those are present in male cone. So here in female cone megasporophylls will be present, and the ovules which are born on those megasporophyll which may be clustered to form the female cone. And here one more thing it has been told, in case of each and every megasporophyll, the upper part, that is the sterile part, also we have discussed in case of male cone, that is commonly known as apophysis. Middle part of the megasporophyll, that is called, or that is containing ovuliferous scale, and the lower part is actually containing the bracts scale. So if you see any of the single megasporophyll, you will see that at the apical portion that is known as apophysis, the middle portion is known as ovuliferous scale, and the basal portion is known as bract scale. Here megaspore mother cell is present. In case of the male cone, microspore mother cell was there. So here megaspore mother cell automatically it will come, and that is divides meiotically to form megaspores. Obviously in that case microspore has been formed, so obviously in this case, that is in female cone, megaspore will be formed. So this is overall the structural difference in case of male cone and female cone, and keep it in your mind, the size or the structure of the female cone will be larger than the male cone even. Male cone will be smaller, but the female cone will be larger compared to the male cone. Going for the next slide, here we will be discussing about the germination of the pollen grain in case of gymnosperms. First, the pollen grain starts germinating with microsporangium and produces partially developed male gametophyte. So pollen grain, it is actually germinating within the microsporangium and producing what? Partially developed male gametophyte, because microsporangium obviously is containing microspores, and microspores will be always giving birth of the male gametophyte. Male gametophyte consisting of two disintegrating male prothallal cells. Keep it in your mind here. Male gametophyte, that will be always consisting two type of male prothallal cells. Those are disintegrating, not integrating with each other, and that is the generative cell. Another one is a tube cell, that is a vegetative cell. We know as present in the angiosperm, there are two type of the prothallal cells, that is vegetative cell and generative cell. So same thing here it is written. Tube cell, that is a vegetative cell, another one is generative cell. Germinating pollen grain are carried by the wind to the female cone. That means anemophily is the process. That means through the wind, the pollen grain will be getting transferred or carried to the female cone. Here there is no stigma structure, no style structure. Here male cone and female cone both are present. The pollen tube produced by the pollen grain entering the tissue of the ovule. So the ovule which is present on the surface of megasporophyll, already I have told in the previous slide. So the pollen grains will be getting transferred on the surface of the apical surface of the ovules, and from there the pollen tube is getting formed, and that pollen tube is entering within the ovule. Pollen tube contains tube nucleus, stalk nucleus and two male gamete. So tube nucleus is also there. Another more nucleus is added here, that is not present in angiospermic plants, that is the stalk nucleus, and two male gametes which are produced from the generative cell, those are also present. The stalk and tube nuclei, they will disintegrate when the pollen tube is entering within the ovule, because these two nuclei have no such function during the fertilization process. Only guiding those male gametes and providing the nourishment is the main function of this stalk nuclei and tube nuclei. So they will get disintegrated, destroyed. The pollen tube discharges the male nuclei or the male gametes, two male gametes within the embryo sac of the ovule, such as you can see in the figure. Like that it is happening in angiospermic plants. One of the male gamete fuses with the egg or ovule to form up the zygote, and another male gamete, as usual, they will be fertilizing with polar nuclei cells to form further endosperm. So this is overall the germination process of the pollen grain. This process is also found in case of angiosperm, but only the different is, in case of angiosperm, the male part, that is commonly known as androecium, and that is containing anther from where the pollen grains are producing. In case of gymnosperm, the male cone is taking entire responsibility for producing the pollen grains containing two male gametes. And in case of, if you consider the female, in case of angiosperm, obviously they are containing stigma, style and ovary. Those are the three parts present within the gynoecium, and here the female cone is taking that charge. And on the surface of the female cone, megasporophylls are present, and those are having those spore sacs, ovules, and through the ovule surface, through the micropylar aperture, the pollen tube will enter. And one more thing, here one more nucleus is actually present within the pollen tube or passing through the pollen tube, that is stalk nucleus, which is not present commonly in case of angiosperm. So I think the germination process of the pollen grain in gymnosperm, it is very much clear to each and everyone. I have come to show you some of the figures. This is the cycas, or it can be considered as Cycadopsida. Pinus, that is another plant species that is belonging from the category Coniferopsida, and Ginkgo, that is belonging from the category Ginkgopsida. So there are three classes or three categories, subclasses present within the Gymnosperm, that is Cycadopsida containing cycas, Coniferopsida, that is containing Pinus roxburghi, which is commonly found in the mountain area, and Ginkgopsida, that is containing Ginkgo biloba. So this is the Ginkgo plant, Ginkgo leaves. So this is overall the three different plants representing the Gymnosperm. That is very much getting clear. Going for the last class, and that is Angiosperm. In the Angiosperm we will be discussing about same, the special features. They are the flower bearing plants. Seed also is present and that is not exposed, obviously remain covered within the fruit. They can be hydrophytes, they can be xerophytes. That means they can be present in the water environment or aquatic environment, as well as they can be present in desert environment. Flowers are having 4 holes: sepal, that is a unit of calyx; petal, that is a unit of corolla; carpel, that is a unit of gynoecium; and stamen, that is the also known as androecium. Ovule is also there, that is a megasporangium, that is enclosed by 1 or 2 integuments. They are having 4 megaspore, that's why it is megaspore tetrad, and each and every megaspore is haploid. And keep it in your mind, out of 4 megaspore, only 3 remain deactivated, or 3 got degenerated, only 1 remain active, and that will be further forming the embryo sac. And keep it in your mind, within the embryo sac 8 nuclei and 7 cells are present. Whatever the 8 nuclei, egg cell nuclei towards the micropylar end. On the both sides of the egg cell nucleus there is a presence of synergid cell nuclei. If you go to the chalazal end, then antipodal cell nuclei, and in centrally located 2 polar nuclei. So total, if you calculate 3 plus 3 plus 2, 8. And if you calculate the cell, obviously antipodal cells towards the chalazal end 3, egg cell towards the micropylar end, and 2 synergid cells, that is total 3. So 3 plus 3, 6. And in middle, polar nuclei, 2 polar nuclei is surrounded by a single plasma membrane, that means it is present within a single cell, that is total 7 cells. Pollination I am talking about, there are 2 types of pollinations commonly found. One is self pollination, another one is cross pollination, that we all know. Self pollination is always happening in the same species or the same plant body, obviously. Cross pollination in same species but different plant body, we all know, because already in the sexual reproduction in flowering plants, that specific chapter we will be discussing in further in class 12 in that specific chapter in a elaborate manner. Egg apparatus, that is containing these portions, 2 synergids at the lower end, that means towards the micropylar end, 1 egg which is present centrally, and that is also towards the micropylar end. 3 antipodal cells, that is present towards upper terminal, that means towards the chalazal end. 2 polar nuclei which are present centrally. Okay, so this is overall the structure of embryo sac or egg apparatus. Going for next slide. Next slide is cotyledon. They are food storing leaf-like structure. Keep it in your mind, the cotyledons can be single or 2, and based on that we have classified the angiosperms into monocotyledonous and dicotyledonous plant. Dicotyledonous plants, they are having 2 cotyledons in their seed, and monocotyledonous plants are having single cotyledon. Dicotyledonous plant, they are having 2 cotyledons, so for that reason endosperm content is very less or sometimes absent. But in case of monocotyledonous, only single cotyledon is present, so for that reason endosperm content is quite high compared to the dicotyledonous plant. In case of dicotyledonous plant, the root system is taproot system or adventitious root system, and in case of monocotyledonous, only adventitious root system is present. The dicotyledonous plant, in case of that, the leaves are quite more containing broader surface. In case of monocotyledonous plant, the leaves are not so much broader, rather they are blade like, narrow. In case of dicotyledonous plant, the stomata are present on the lower surface only, or monocotyledonous plant, the stomata will be properly divided or present properly on the both of the surfaces, not only confined in the lower surface. In case of dicotyledonous plant, we all know the venation which is found, that is the reticulate venation, and in case of monocotyledonous plant, obviously the venation type which we are commonly found, that is the parallel venation. So these are the overall differences which can be found in the dicotyledonous plant and monocotyledonous, in between dicotyledonous and monocotyledonous plant. So, I will be discussing these differences also next. Plant life cycle, that is containing both of the phases, gametophytic phase and sporophytic phase. Gametophytic phases means those are always containing haploid generation, and sporophytic phase is always diploid generation. Keep it in your mind, plant life cycle can be isomorphic and heteromorphic. How? Isomorphic means gametophytic and sporophytic phases both are similar looking, that's why they are known as isomorphic life cycle. In case of some of the angiospermic plant, the life cycle can be heteromorphic also, because morphologically both of the sporophyte and gametophytes are not similar, rather different from each other. So these are overall the basic and specialized features present in case of angiosperms. Going for the double fertilization procedure, which is very much important, only found in case of angiosperm. First of all, the anther of the stamen, that is producing the pollen grain. By the process of pollination, that pollen grain will be getting transferred to the surface of the stigma of the carpel. Then the pollen grain will be obtaining the nutrition, and its intine layer, it will start forming the pollen tube, and that pollen tube will be penetrating through the surface of stigma. Keep it in your mind, through this pollen tube, 3 nuclei are passing. One is tube nucleus, another one is 2 male gametes. So before entering through the micropyle of the embryo sac, the 2 male gametes, obviously they will be entering within the embryo sac, but before that the tube nucleus is totally get degenerated. How it is happening in case of gymnosperm? The same manner. Keep it in your mind, the 2 male gametes which are actually entering within the ovule, within that the first male gamete, that is entering within the ovule, embryo sac rather, and fertilizing with the egg to form the zygote, and another male gamete, that is reaching to the polar nuclei centrally located, and by fertilizing with that, it is forming triploid endosperm nucleus or PEN or primary endosperm nucleus or endosperm, that is triploid number of chromosome containing. So this is the overall double fertilization mechanism. The difference in between the double fertilization mechanism in gymnosperm and angiosperm I have already told you when I have discussed about the gymnosperm, because in gymnosperm there is no anther, there is no stigma, there is male cone and female cone, and remaining entire the procedure whatever I have just discussed, that is entirely same. Now I am just moving for the discussion about the life cycle of angiosperms. If you see the life cycle of the angiosperms, always this is the plant which is commonly found in the environment, that is a sporophytic plant body. So in case of angiosperm, whatever the plants commonly we can see in the environment, those are all present in the sporophytic phase. Sporophytic plant body, obviously they are having stigma, okay, because stigma is a part of the female reproductive system, and they are having also the male part, that is androecium. So always keep it in your mind, they are having microsporangium, that is the pollen sac which is present within the anther. This microsporangium is containing microspore mother cell, from where the microspore or the pollen grains are taking birth, and for that obviously meiosis cell division is required. In female reproductive system, they are having megaspore mother cell, and they are also having megasporangium. So the megaspore mother cell will be further dividing meiotically to form the megaspore, that is embryo sac. Here always keep it in your mind, embryo sac always will be 1 in number, it will be produced, because actually during the formation of the embryo from megaspore mother cell, there are also 4 megaspores are getting produced, but out of that 4 megaspores, 3 are getting degenerated, only 1 remain activated. And in case of microsporangium, from the microsporangium where the microspores are getting produced, 4 microspores can be produced at a time from single microspore mother cell present within pollen sac. So now the microspore or the pollen grain, they are having 2 cells within them, vegetative cell and generative cell, and that is getting produced by the mitosis cell division happening within the pollen grain. Now pollen grain is getting transferred on the surface of stigma. From the stigma surface, it will be start entering through the pollen tube, and in the pollen tube both of the male gametes are following actually one specific cell or nucleus, that is a tube nucleus or vegetative cell nucleus. Now both of the gametes enters within the embryo sac. This is the structure of embryo sac. It is entering through which? Through micropylar aperture. Just now I have told in the previous slide. Then when the first male gamete is fertilizing with the egg, automatically it is forming the zygote, and another male gamete is fertilizing with the polar nuclei to form the endosperm. But we will not be discussing about the endosperm because endosperm is not our topic actually. Our topic is to complete the life cycle. So after formation of the zygote, so this zygote is further getting developed into the embryo. Initially it is forming pro-embryo, then it is forming globular shaped embryo, then it is forming heart shaped embryo, and like that, and so on. And then it will be forming a full plant, that is sporophytic plant body. That means this cycle, if you see clearly, the yellow portion of this cycle, this portion I am talking about, this entirely the sporophytic plant body or the sporophytic plant generation, that is the 2n generation, and this portion is only the haploid gametophytic generation. So this is the sporophytic generation, this is the gametophytic generation, whatever it can be found, because their life cycle is getting differentiated into 2 generations. Next, we are going for discussing about the plant life cycles or alternation of generation. What is that? Plant life cycle or alternation of generation, that means we have already discussed now that in case of throughout the plant life cycle, if you see, there are 2 phases or 2 generations can be found. That one is sporophytic generation, another one is gametophytic generation, and this sporophytic and gametophytic generation continuously they are coming one by another. After gametophytic generation, sporophytic it will come, after sporophytic gametophytic will become, so they are coming one by one and continuously. So this phenomenon is commonly known as alternation of generation or metagenesis. So let's see whatever the types of life cycles commonly found in case of plants. Mainly there are 3 types of the life cycles commonly found. One is haplontic life cycle, second one is diplontic life cycle, and third one is haplodiplontic life cycle. So let's start with the haplontic life cycle. From the name we can understand the haploid generation is the maximum, it is taking the maximum time, or it is the dominant generation. So that's why the life cycle has been named haplontic life cycle. Keep it in your mind, this is a single phase. Dominant generation is obviously gametophytic because the name has been given haplontic life cycle, that means the gametophytic generation will be the dominant generation. No such alternation of generation can be found because haplontic to diplontic it will not happen or it will never happen. And this haplontic life cycle is commonly found in case of algae or Pteridophyta. Start seeing the cycle. Suppose this is the gametophytic phase of the plant having the n number of chromosome. It is forming the male and female gamete by the mitosis cell division. So for that reason male and female gamete is having also the haploid number of chromosome. Now the male and female gamete they are fertilizing with each other and the syngamy has happened, and after that they have formed zygote, that is 12 number of diploid number of chromosome containing. This zygote will be getting further passing through the meiosis cell division, and just it will be passing through the meiosis cell division, again the meiospores will be getting produced, and these spores when they will germinate automatically the gametophytes again will be formed. Okay, so that means throughout the entire cycle you can see one thing, throughout the entire cycle most of the stages or most of the phases are actually haploid number of chromosome containing, rather diploid. Only this specific phase or this specific stage zygote is only the diploid phase. So obviously it is getting clear to each and every one that throughout their entire life the haplontic phase or the gametophytic generation is actually remain dominant. So for that reason this is commonly known as haplontic life cycle. And keep it in your mind, which plants are showing this? Algae and Pteridophyta, they are showing haplontic life cycle. Going for the diplontic life cycle, 2 phases are found, that's why it is the diplontic. Dominant generation is sporophytic, obviously. Alternation of generation is found and commonly found in the Gymnosperm and Angiosperm. So let's see how it is actually showing both of the generations, both of the phases. First one, that is the sporophytic phase, that is diploid number of chromosome containing. The sporophytic phase is further producing their gametes, that is the gametogenesis process, and obviously during the gametogenesis the meiosis cell division takes place, and that is forming the male and female gamete, that is haploid number of chromosome. This male and female gamete is fertilizing with each other and obviously forming the zygote, that is the diploid number of chromosome containing phase or stage. This zygote will be further getting converted into sporophytic generation, that is 2n number of chromosome. That means throughout this entire cycle you have seen, except this male and female gamete, remaining stages, that is zygote and sporophytic plant body, they are containing diploid number of chromosome. That means mostly they are showing their diploid phase. Only one stage is haploid. That means the sporophytic phase is getting dominant in this life cycle. So for that reason, this life cycle is commonly known as diplontic life cycle, found in Gymnosperm and Angiosperm. Came to the last life cycle, that is haplodiplontic life cycle. That means haploid and diploid, both of those generations can be found properly in this life cycle and found in thallophytes, bryophytes and pteridophytes. Both generations are present separately and length of the generations are almost equal. So let's see, sporophytic plant body, 2n number of chromosome. Sporogenesis has occurred, that means the spores are forming. Those spores are further moving through the meiosis cell division and forming meiospores, and those spores are further germinating to form the gametophytic plant body, that is haploid number of chromosome containing, and that haploid number of chromosome containing gametophyte is further producing male and female gamete, and male and female gamete obviously it will be fertilizing with each other and forming the zygote, and that zygote will be further getting developed into sporophytic plant body. That means sporophytic plant body is diploid number of chromosome containing stage. When it is present in the before sporogenesis, up to that they are showing diploid number of chromosome containing structure. After meiosis it is getting haploid, that means in the gametophytic generation it is getting haploid. Male and female they are getting haploid. Just they are again forming zygote by the fertilizing with each other, they are getting again diploid. That means two phase or two stages, sporophytic stage and zygote stage, they are diploid, and gametophytic stage and meiospore or male female gamete they are haploid. That means throughout the life cycle you can see both of the male gametophyte and sporophytic stage or haploid or diploid stage equal or equally 50-50 almost. So for that reason this specific cycle name we have given haplodiplontic life cycle, found in Thallophyta, Bryophyta, Pteridophyta, that means algae, moss, bird. So this is overall the types of life cycles and found in different plants. These specific cycles are also drawn in your NCERT book, you can see that, but in a different manner, in a circular manner it has been drawn. So if you think or if you see that will be getting very much easier for you to draw, to remember, you can also follow that, or if you are thinking that this one is getting easier to you, you can follow this one, or if you can remember this one also. Both are similar. This is only a box like manner I have represented, they have represented only in a circular manner. That is the difference mainly. But I think the concept is very much clear to each and everyone, and with this specific slide, haplodiplontic life cycle, the entire video on the plant kingdom, specifically on the Pteridophyta, Gymnosperm, Angiosperm, it is getting finished, and I think everyone has enjoyed this video a lot. Thank you everyone for listening this video. Thanks a lot.